Hello, I'm Christina Laukaitis, MD, PhD. I'm an associate professor at the University of Arizona. Greetings from sunny, hot Arizona. I'll be speaking about genetics for beginners. At the end of this talk, I hope that you will understand four things. One, that genes are written in the language of DNA and bundled into chromosomes. Two, genes are the instructions for the jobs that the body needs to do. Three, genes are passed from parent to children. And four, when genes are misspelled, diseases can occur. So I'm going to start big and focus in from organisms all the way to genes. So there's a few basic concepts that are probably reviewed for most people, but in case they're not, I'll introduce them in order that we can all understand the terminology and the words. So a person's body is made up of multiple organs that do jobs. For example, the brain is important in helping people to think and process information. The kidneys filter the blood. The intestines digest and absorb food. Organs are made up of tissues. So an organism has organ systems. This is the digestive system. And these are made up of organs like the stomach. The lining of the stomach is a tissue, and this tissue is made up of multiple little cells. As I said, tissues are composed of cells, and these are four different types of tissues. In this epithelial tissue, it's quite easy to see the cells. This is a cell outline and a cell nucleus. Nervous tissue has very clear cells with a lot of connections. Muscle cells are harder to see. They form a network. Connective tissues are unique. Connective tissues are important in holding together other different tissue types. They're especially important in the skin and in the lining of the gut and in the blood vessels, but they're really important in many tissue types because they hold things together. But like other tissue types, they are composed of cells. You can see the cells. They also have a lot of stuff around them, a lot of proteins or matrix. And we'll talk about what proteins are in a few minutes. Cells are all important for the jobs of the body and the jobs that the tissues and organs do. And the instructions that tell a cell what to do are located in the nucleus. So this is a big blow up of a cell. We've gone from whole organisms to tiny little parts. And even within this tiny little part, there are subsections. There's a nucleus where instructions or DNA is held. There are also other things, we call them organelles, which do other jobs within a cell. That lets a cell do its job within a tissue, that lets a tissue do its job within an organ, and an organ do a job within the body. Finally, to go very micro, once you go from cells to nucleus, within a nucleus, we see chromosomes. And chromosomes are big lines of DNA. And so DNA is organized into chromosomes. There are 3 billion bases of DNA in the nucleus of every cell of a human body, and they have to be organized. And so they're organized into these structures called chromosomes. The DNA is what the genes are written in, and we'll come back a little bit more to genes later on, but let's talk chromosomes for just a second. This is a schematic. It's actually a picture of a person's chromosomes. Usually chromosomes don't look nearly this neat. Most of the time, chromosomes are spread out and spewed through the nucleus. And they're organized more than you might think. They're organized based on what's most active in a cell at one time. I think of the nucleus a little bit like someone's closet in their home, and the chromosomes are all of the clothing, or actually the genes are all of the clothing in your closet, and so they're just spewed wherever. But there's usually even some organization in a teenager's room. Those clothing that get worn more often or more actively in use usually rise to the top of the piles. Those things that are not being used very often get pushed toward the back. And that's what happens in a cell. The genes that are most active at any one time get put in places where they can be accessed easily. Those that are inactive get pushed back. Only when cells duplicate their DNA and get ready to divide into 2 cells do we see things this neatly organized. And this is even neater than it occurs then, because someone has taken a picture of a nucleus and cut the chromosomes out to put them next to each other. These banding patterns, these dark and light spots, have to do with how much protein is around the DNA and how accessible that DNA is. There are two kinds of chromosomes. There are chromosomes called autosomes, which are chromosomes that can be inherited either from your mother or your father. And then there are two special chromosomes, the sex chromosomes, the X and the Y. Here you get X's from mom or dad, but you can only get a Y from your father. And so these are the special ones that determine a person's gender. Overall, people have 46 chromosomes found in 23 pairs. At least that is the typical state. So genes are written in the language of DNA and bundled into chromosomes. Genes are also the instruction for a body's job. So again, here's our schematic cell with a nucleus, that's yellow, and within the nucleus is the genome. So the genome is the term for all of a person's DNA. It's all 3 billion plus pairs of DNA. As I said, it's organized into chromosomes, and these chromosomes are just big bundles of DNA packaged up together. And the DNA then has genes. So sections of this DNA encode genes, or are genes. And the genes are basically the instructions for making proteins. Okay, let's talk about proteins. Proteins are the things that do work at a very tiny level in the body. Some of them are enzymes, like the enzymes that are in our intestine that help to digest food and allow us to absorb it. Others make structural proteins, things like collagens. We've all heard of collagens here. They're very important in connective tissue, and when they're not made typically, can lead people to have hypermobility and different types of EDS. So these are both examples of proteins. And so proteins are what genes are the instructions for. This is a different way. DNA are the instructions. RNA is I'm not going to talk much about, it's just a messenger that leads to proteins. And proteins are the body's workers. They're what does the day-to-day job. This is so important in biology, we call it a central dogma. The central dogma of biology is that DNA goes through an RNA intermediate to make protein. And this is the basic point of molecular biology. So, as I said, there's a central dogma where DNA goes through an RNA intermediate to make protein, and that is what causes a person to be able to function, and it changes the physiology of a person. When there's a problem at the DNA level, it gets carried through each of these other levels and can lead to differences in protein and differences in a person, differences in physiology. So, back to our outline, genes are the instructions for the jobs of the body. Now let's talk about genetics and passing on genes. So a couple of important key things to know here. Every person has 3 billion bases of DNA in its nucleus, or every person's cell in each nucleus has 3 billion bases of DNA. And every time a cell divides, it has to give an exact copy of that DNA to the daughter cell. So before any cell can divide, it has to copy all 3 billion bases of DNA. This means that every cell starts with 2 copies of each gene and then duplicates it to end up with 4 copies of each gene, and then divides so that each daughter cell gets 2 exact copies. This whole process of copying is quite complicated. It works because of the double helix nature of DNA, and that's its own set of lectures, but it works because G's always bind to C's and T's always bind to A's, and it just works. But sometimes it doesn't work perfectly. Mistakes can happen. There are 2 different types of cells in a person's body. One are called somatic cells. These are the cells that result from other cells. They're the cells in your skin or in your stomach lining or in another organ, and these cells have to keep dividing to make up for new cells as cells die. When a mutation happens in the process of making a somatic cell that might have very few implications, or it might have very severe implications. And some of these mutations, when they build up, are exactly what leads to cancer. So mutation is important in cancer, but it's not the kind of mutation that gets passed to future generations, but it still is important in your body as you age. When mutations happen in germ cells, and germ cells are the cells that make up egg and sperm and get passed to the next generation, then these can cause diseases. And so ultimately, mutation comes from somewhere, and usually it comes at some level far enough back from problems with this process of DNA replication and splitting. So in typical cells, somatic cells, the cells of the body, we start out with 46 chromosomes, then there's a duplication, and these have to split in a very organized way to end up with 2 daughter cells that each have 46 chromosomes. So this is what happens in our somatic cells. When this doesn't happen right, sometimes people end up with cancer, sometimes we end up with cells that just don't work well. And in the end of the day, each daughter cell ends up with 2 copies of each gene. So that's the typical process in somatic cells. And it's a process. It doesn't always work perfectly. So sometimes things do go wrong. Hopefully we don't get the lawyers involved. Egg and sperm are different. Germ cells are different. Here, while we start with 2 copies of each gene, there's a set of divisions that occur so that in the end, the sperm or the egg only end up with 1 copy of each gene. So it's a little bit different process, no less complicated, in fact, even more so, but it ends up with this reduction in gene number. Why am I telling you this? Because it's responsible for this genetic process we call segregation. So I'm going to introduce some nomenclature here because you'll hear it. And so you want to know where it comes from. So different versions of a gene are called alleles. And so most people have, everyone has 2 copies of each gene. And if they are slightly different from each other, then they're called different alleles. So this process of making germ cells, where we take 2 copies of each gene and end up with only 1 in the sperm or only 1 in the egg, is a random process. So it is a process called segregation, and each time a sperm is made, whether it gets the big R from this, actually from this mother, or the little r from this mother, then this is a 50-50 proposition. So the germ cell, progenitor germ cell, and has both gene copies. In the process of going through meiosis, then it makes eggs that have just one or the other. The same happens if sperm has 2 different alleles, as sperm from a father with 2 different alleles. Either the big R or the little r gets bundled. And then when those come back together, here are the 4 possible children of this coin flipping father and coin flipping mother. So this brings us back then to Mendel. So I don't know how many people in this room are currently having nightmares of high school biology. You're allowed, I will acknowledge that. This is a really bad drawing of Gregor Mendel, but it shows his pea plants. And this is what Gregor Mendel's insight was, that segregation of different alleles causes recognizable inheritance. Mendel was able to look quantitatively at genetics and understand that this wasn't a blending process, it was a separation and rejoining issue. So here are our pure breeding yellow peas and our pure breeding green peas that come back together. And then when those 2 meet with each other, we're able to see the effects of segregation in the 2nd generation. Once people are able to travel again, I'll put in a small plug for the Mendelianum in Brno, which is an excellent historical and modern museum devoted to Mendel and his work. So let's come back to humans. Enough about peas. In human terms, all of this is what leads to our major inheritance patterns of autosomal dominance and autosomal recessive inheritance. So let's talk autosomal dominant first. So here we have 2 parents. One doesn't have the disease that we're interested in, and the other does. Each of these has the typical normal case of having 2 copies of this gene. So we show 2 chromosomes here, one for dad's pair chromosomes, one for moms. Each has 2 copies of the gene of interest. In Dad's case, one of his copies carries a mutation. So Dad has 2 different alleles. One allele has a mutation. This allele with the mutation dominates over the normal copy, causing Dad to have a disease. Mom still has 2 copies. It's just that both of her alleles are not disease-causing. They're normal. So what does this mean for the next generation? Well, as I said, when Dad bundles up sperm, half of his sperm will get the allele that causes disease. Half of his sperm will get the allele, the gene copy, that does not cause disease. Any children that are made with the sperm with the disease-causing allele will also have this dominant allele and have disease like Dad. Any children made with the sperm that doesn't contain the disease-causing allele, the disease-causing gene copy, will also get their normal copy from their mom and not have disease. So half of the children of someone with an autosomal dominant condition will likewise have that condition. Everyone, both parents and children, always has 2 copies of the gene. It's just that some of the one parent and half of the children will have one of those copies cause disease, or one be a disease-causing allele. This is a little bit different than autosomal recessive inheritance. Autosomal recessive inheritance is caused when a person has 2 disease-causing alleles. In the case of autosomal recessive inheritance, parents or people with a single copy of a mutation don't show the disease. That's because this disease-causing allele is recessive to the normal allele. So the normal allele here is dominant. The disease-causing allele is recessive, and thus people with a single copy of a disease-causing recessive allele have a normal phenotype. They don't have disease. So a genotype is what the genes look like, and a phenotype is how a person looks. Does the person have disease or not? So children of people who are carriers for an autosomal recessive allele can either have disease or not have disease, but in a little bit different proportion than with an autosomal dominant condition. So for a child of someone who's a carrier to have the disease, that child has to have been made with a sperm that has the mutation and an egg that has the mutation. Both of those events have to happen for this affected child to be made. And that would happen statistically 25% of the time, in 25% of the offspring of 2 carrier parents. If one parent gives the child a disease-causing allele and the other gives the child a normal allele, then that child is a carrier but doesn't show any signs of disease. And this would happen half of the time. Finally, it's also possible that this child is made with sperm that have a normal non-disease-causing allele and egg that has a normal non-disease-causing allele, in which case that child is unaffected and not a carrier, so can't pass that on. So 25% of children of someone with an autosomal of 2 parents who are carriers of an autosomal recessive condition will be affected. Half will be carriers and unaffected. 1/4, 25%, will be non-carriers and non-affected. If one parent is a carrier and the other has a wild type genotype, then they will not have any children who are affected. There is a third kind of inheritance, and that's sex-linked inheritance. Here, these are what happens when genes are encoded on the X chromosome that are and are disease-causing. So for X-linked or sex-linked inheritance, it matters which parent is carrying the disease-causing allele. And this is really more complicated and not particularly relevant for EDS. So I'm not going to go into this. So we've talked about genes being passed from parents to children. Now let's talk a little bit about mutation. I've had to assume that people understand that gene mutation leads to disease, but let me talk now about how that happens. So this will be a bit more mechanistic. And then as I understand it, the next talk we'll talk even more in detail about this. So as I said, DNA is the language of our genes, and this is our genetic code. The way this language is structured is that there are 3 DNA bases, 3 triplets of A's, T's, C's, and G's that each code for an amino acid of a protein. So as I said, the central dogma is DNA, RNA, protein. The DNA language is triplets. The protein language is these amino acids. And so DNA language is the A's, C's, T's, and G's, and the words are organized as triplets. And these code for proteins, which are made up of amino acids. All right, so what's an amino acid? I think amino acids a little bit like Legos. These are the parts of the structures that make things up. And so different triplets of DNA each encode for a different Lego type. TTT encodes for phenylalanine, which is, our yellow 8-top, whereas CTT encodes for leucine, which might be a green 4-top. And putting these in a certain order then is what are the instructions for making whatever it is you're making. And just like with Legos, the body makes any infinite number of different proteins. So proteins are made up of amino acids that are encoded by codons of DNA. A few of these amino acids are special. These are what are called termination amino acids or terminators. These just tell the Lego structure, they tell the protein to quit being made. It says, you're done, move on. So there are some special terminators here as well. So DNA mutations can change the protein. So if there is a difference in DNA, you can it'll encode and say, use a different amino acid, use a different Lego, and make a different protein. In this case, I'm showing an example where the DNA words are CAT, CAT, CAT. Each of these encodes a histidine, and so you get this run of histidines as your protein, a bunch of white 8s right in a row. But if you exchange this T for this A, then suddenly you've thrown a glutamine in there, and a glutamine is a black 6. So you get a bunch of whites and then black and then whites. So you get a difference in the protein. And Dr. Malfate's going to talk about the relevance of those differences. Sometimes they're extremely relevant, sometimes not so much. Sorting through that is challenging. There are other kinds of mutations, though, that are much more often problematic. And these are things that are frame-shift mutations. As I said, the language of DNA is triplets. The words are 3 letters long. Hiss, 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 by cat, 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 cat. But if you cut out one of these, if you cut out that C, then suddenly you change the way the words are structured. And so cat, cat, cat goes to something completely different. Cat, cat becomes isle, 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 and then that'll continue until the protein hits a stop codon and the whole thing ends. So basically, you go from Lego structure to Lego chaos, and then the protein ends. And so that is a much more disruptive and problematic kind of mutation. I can go on about kinds of mutation all day. Again, it's a whole subfield of genetics, but you're just here for the basics. Let's bring this back then to what you're here for, which is EDS. I'm going to spend just a minute talking through the nomenclature of and the way that mutations are named so that you understand. If anyone's had genetic testing and had something found, you're going to get a report back that looks like a bunch of letters and random letters and random numbers, but there's meaning to this. So these are examples of 3 different mutations in collagen 5A1. These 3 different mutations cause classical EDS. So in the top one, this is the collagen 5A1 change, and this is a specific substitution, position 4916. A G is supposed to be there, and it's changed for a C. What happens is this C for a G change causes a cysteine to change to a serine. So it causes a black Lego to change to a yellow Lego. And this, in this case, is just in the right place to cause classical EDS. It's actually one of the rarer mutations, for rare point mutations that does cause, or rare substitution point mutations that causes classical EDS. This one does. A second mutation type is this second one, and here again in collagen 5A1, a substitution of a T for a C at position 265 causes a glutamine amino acid to be changed to a terminator codon. So here, the Lego ends. You make no more protein because this glutamine that was early on is substituted for something that says just stop the protein right there. So it ends right there. This is an example of a frame shift mutation where at position between 226 and 227, there's a 1 base pair insertion. This causes an arginine to, a frame shift to be made at an arginine. So you get 76 more bases of gobbledygook, of garbage, and then you get a stop codon, protein ends. So this is how mutations occur and how the nomenclature, or how we label them, and how you understand that. So, genetics for beginners. You now understand about genes and DNA and chromosomes. You understand how genes are the instructions for doing the body's jobs and how they're passed from parents to children. And finally, I've told you a little bit about how genes encode for proteins and how when that process goes wrong and the genes are mismade or misspelled, diseases can happen. So now that you're all experts in DNA, I'm going to hope that you get out there and start working and start making some clones. Any case, you can ask questions at the scheduled question and answer session, and I hope you all have a very good day. Bye-bye.